Okay, so welcome to this first video for National 5 and Higher Biology Revision. This is just a little introduction on how to revise the best methods of revision and also to show you our National 5 and Higher Biology Revision Google Classrooms. So as you can see, I'm on the National 5 Biology Revision Google Classroom just now. I always think it's easy to go to classwork view because it lays it out by topic. And as you can see at the top of the page there, just like the higher biology revision one, which I'll show you in a moment, it says please read this first at the top. So I'm just going to click on that. And this talks about the 4 categories of resources that are available on this Google Classroom. These are learn, check, test and skills. And the idea is that you progress through these 4 different stages, if you like, especially 1, 2 and 3 for knowledge. Skills is a little bit separate, so I'll talk about that later. But for the learn, check and test, this is for the topics, the key areas of the course, the knowledge that you have been working on. And we're now at a stage in the year where we're preparing for the prelim. So we've already completed at least 1 unit. We've almost finished unit 2 in most courses, and if you're in a 2-year higher, you'll have done a lot more than that as well. So for the learn topics, first of all, let's have a look at them. So these are summaries, videos and other resources which allow you to go over topics that you've missed or you found difficult in class. If you had done your Unit 1 assessments, your unit assessment and your ABC assessment, and you had a key area that you did really well with, you scored well in that section according to your assessment analysis, you wouldn't bother with the learn section because this is really for topics that you found tricky and you need to go over them again. It's the same with check. Check are simple, straightforward questions or ways of testing your knowledge at a kind of C level, if you like. And again, these are straightforward questions and activities designed to build your confidence. So again, if you've done really well in your Unit 1 assessments, I should say, then you wouldn't really want to do the check questions, for those topics. However, if you were not confident with a particular key area, again, based on your assessment analysis, you would do a little bit of learn, a little bit of check, and before going on to the test. So the test section, and I'll show you all the details of these in just a moment, the test section is all exam standard questions. These are past papers, sometimes they're just past papers themselves, sometimes they're organised into individual units, and even down to individual key areas. You can go straight to these for topics that you're confident with, so ones that where you, according to your assessment analysis, you've done well with. Or if it's ones you're less confident with, you'll have gone through the learn and check sections first. So look at the learn activities first. There's many versions of the PowerPoints, called Crash Course PowerPoints. There's a Matrix of Knowledge, and I would always refer back to that. That gives you the correct terminology for all the topics and shows you exactly what you should know for each key area. You've got YouTube videos for revision, which are really excellent. And again, they're both, as you'll see in a moment, they're available for hire as well. And we've also got summary notes. Okay, so if I click on the Unit 1 summary notes, for example, you can see that it covers all the different key areas as separate topics. And for other units, I think Unit 2, it's all together. Okay, so either way it covers them key area by key area, or it's the full unit, again with chapters in it. You can see that Unit 3 is greyed out at the moment, and that's because none of our National 5 classes have yet gone to Unit 3. I'm looking at the teacher view here, so you guys wouldn't see that. For the check section, remember this is questions that are pretty straightforward. It's either getting the basics for the terminology or some of the key fundamentals of the course. Important if it's a topic you've struggled with. We've got Quizlets, quizzes, we've got Google form quizzes as well on each of the key areas for Unit 1. And all of these have got the answers with them. You can check your answers. Quizlet I really like because it gives you Quizzes I really like because it gives you the feedback for your questions as well. It tells you ones areas that you need to work on. And if there's questions that you're continually getting wrong, Quizzes we'll keep going over those until you're getting them right. Okay, Quizlet's excellent for definitions, and the Google form quizzes are great as well because they mark themselves and you can use that to feed back. Going on finally to the tests section for knowledge. There's a really good site called Achieve Learning, and you can get the login details for that from your teacher. The one I really like though is the past paper questions by key area with marking schemes and course specs. I'm just going to click on that second one down just now. And there's a number of things I like about this. First of all, it lays out the topics. So you're now at the stage where you're ready to really test yourself at exam level. Here's, for example, cell ultrastructure. Each section, it gives you a little reminder of what's in that topic. You'll then click on revision for 1.1 once you've had a wee read over that, reminded yourself what's in it. And you simply get lots and lots of past paper questions by topic. Okay, so you could either type these up on a document, Google Doc, or simply write the answers down. And once you've finished all of those questions, some are multiple choice, some are short answer, at the bottom you can see there's a marking scheme that will open up a different Google Doc which gives you all the answers. Okay, so you can check your work and see how you've done. So I really like that resource. It's really excellent for National 5. There's a similar version for higher, which we'll show you in a second, but that's really an excellent resource. And we've obviously got the actual past papers themselves, and we're going back quite far. We're going back to 2014. These ones are not available on the SQA website, so that's really useful as well. These multiple choice past paper Google Forms are for whole units. So the difference between these and the check section is there by key area, the Google Form quizzes, whereas the multiple choice and the test section are past paper again, but this time for the whole topic. Okay, and we've got lots of other bits and bobs there you can see. Skills is a bit different, and we'll do a video later on in skills, so don't want to dwell too long on that, but as you can see there's parts of this which involve you teaching you how to do the skills, and there's lots of example questions as well. Let's go on now and look at the higher biology revision Google Classroom. I won't spend too long on this because it's very, very similar to what you've just seen for National 5. Again, it automatically opens up in the view that says Stream down the bottom. So if you go to Classwork View, again you'll get it topic by topic. And again, it's the same setup. We've got Learn, Check and Test. Some slight differences. The summary notes I really like because, I really like the summary notes because they have got check test questions as well. So I'll just show you that once it loads up. Apologies for the delay there. So this is the summary notes here, and this is for Unit 1, DNA and the Genome, which by now in the course, this stage of the course, we've all done, so it's a good one to show you. You can see the contents, but at the end of each chapter, some nice notes and diagrams. There's concept check questions, which are similar to our key area questions that you'll be doing in class. But again, if I go back here, the other document that's there will give you the answers to these as well. So it's really a useful resource. And again, as with National 5, if it was a key area that you were really good at, you wouldn't bother with the learn stuff, you would jump straight to test. Okay, but assuming it's a topic that you want to work on, you can see we've also got higher revision videos there. We've got a bit of higher exam terminology. Some of the words that we use at higher can be quite, the SQA use at higher to ask the questions can be quite tricky to understand what they're asking for. Words like explain, describe and justify. And we've also got a big section there on key processes, big processes that occur in higher that if you can master those, the stages and the order that they go in, then you can gain lots of marks. For the check section, we've got some Quizlets and some Quizzes, and we'll probably put some glossary stuff in there as well. And again, as with National 5, we've got the test bit. So for if you've been through learn and check materials for your key area that you're working on, or if you're already doing well in a particular key area, you'll go straight to the test section. Again, we've got higher past papers by topic. We'd say by key area, of course. And these go from 2015 to 2019 and also 21-22 as well. These work a little bit differently to the National 5 ones. As you can see, it's hyperlinked. So that if I click on a question, that's the blue underlined sections, and I've been studying, for example, key area 1, I'll click on that top question there for 2017. It will take me to, oh sorry, you do need to open up. Sorry, I don't have my PDF there. But if you've opened up with the PDF or in a Google Doc, it will actually hyperlink you to the questions, and then it will take you to the marking scheme as well. So we'll show you that. Okay, and we've got some extended response questions there. Sometimes we call these essays, but extended response is the proper name for them. That's the either 4 or 5 or 8 or 9 markers that you get in the exam. And also, like with National 5, we've got the Unit 1 and Unit 2, because of course we've not done Unit 3 yet. Unit 1 and Unit 2, past paper questions, multiple choice form. So again, you just open that up and it opens up as a just put your email in, put your name in, and then you get to try the questions. And again, as with National 5 one, it will mark it at the end for you, and you can check over the ones you got wrong and do something about those. And again, it's got the link to Achieve, which is also really good for hire, so you can have a look at that website. You can get login details off your class teacher. And finally with skills, again, it's a mixture of how to do the skills, some advice, and also some skills and past paper questions to practice as well. Okay, so we're going to leave it there for this section of the video. We're then in a little while in the next section we're going to look at actually trying some exam standard questions to help you prepare for your prelim. Okay, so welcome to the second part of this video for National 5 Biology. We're going to look at the 2023 paper, and we're going to pick out some of the skills questions. Quite often pupils find these the most challenging questions in the National 5 assessment. So we're going to look at two of the big skills questions in 2023, questions which I've got pretty much all skills, and then if there's time we'll go back and look at a couple of other individual ones that are sort of peppered throughout the paper. But in order to help us with this, we're going to use some resources again from the Revision Google Classroom. So as you can see in the screen here, I've put the National 5 Biology Revision Google Classroom back up, and the last section, and please read this first part, is skills. Okay, skills are a huge focus in National 5, and this topic has got all sorts of guides, questions and marking schemes to help you, so you can look through that at your leisure. If we go just to the skills part, at the bottom there, you can see there's lots of information here, and we're going to use the skills advice. Now you might have this in paper form from your class teacher as a little booklet, so it's called the skills advice booklet, and it just takes us through what we need to know for skills in biology. Here it's presented as a Google slide. So as you can see, it's set into 3 sections. So we divide up the skills in biology. There's questions which are based around experiments and variables. There's questions which involve drawing and interpreting graphs and tables, and there's calculations. And whichever skills question you're doing, there's a little guide to tell you a bit about it. So for example, the first part here, variables, it looks at controls as well. Constant variables, things we need to keep the same in experiments to make them valid, and also looks at the difference in validity, accuracy and reliability. And it's important in biology that you're able to know the difference between those things and how they relate to experiments. In part 2, we look at how to draw a graph, gives good advice about how to draw graphs, and also a little section at the end about describing graphs and tables. Okay, and then the final bit would be like our conclusions, how we explain graphs, what is the significance of the data. So describe a graph, we're just saying what the patterns, what the changes are. When we explain a graph, we're telling us what the information means, usually in a biological sense, so it's quite often related to your subject knowledge. For calculations, we've got a number of different ones that we cover here. Of course, we can't cover all the skills questions, and there'll be some calculations just come up on the day of the exam, which you'll just need to have a go at. But we can help you with averages, average change, which is different to averages, 3 different types of percentages. We've got a section on ratios, and then finally what we call pair questions. Pair means for one of, and you're most familiar with this from miles per hour. When cars drive cars, it's this idea that pair means for one of, and it gives you advice on 2 different types of pair questions. So I'll come back to for the questions that the skills booklet can help us with, I will come back to the skills booklet as we do the questions. So let's start with question 6, which is the first big skills question in this particular year, 2023. This is an investigation which was carried out into GM crops, basically, genetically modified cotton. And we did genetic engineering in section 6, and we come back to it again in unit 3 after the prelim. In this particular study, they've looked at three farms. And each of the farms has got 2 fields of cotton, one with the original and one with the genetically modified. They've recorded the yield. When you see yield in biology, this is basically the harvest, how much in terms of mass of plant or useful part plant that they have managed to harvest. And the results are shown on the table there. Okay, so you can see that the numbers, because yield of cotton is the overarching heading, all these numbers are in kilograms. And the first one is a percentage increase question, which says calculate percentage increase in yield when using modified cotton in farm 2. Okay, so if I jump back just very quickly to our skills booklet, percentages are obviously a type of calculation, so I'm going to go to the third and final section on calculations, and I'm going to find the bit on percentages. Now it's percentage increase, so it's not percentage of a total, It's not percentage when you're given the percentage. This would be an example of a percentage change. And this is one that most commonly comes up in biology. You may see the other two from time to time, but this is definitely the one that comes up most often. And the explanation on the screen there with a reworked example, you can look at that at your leisure again when you're on the Google Classroom. But basically the formula is change in reading over initial. Initial just means the start, the reading at the start, and we times it by 100. So we're going to apply that formula to this particular question. So remember that formula, change over initial, or change over start if you prefer, and we're going to times that by 100. Okay. So the tricky thing sometimes as well in biology is picking out the right numbers. We're only interested here, as you can see, and in the actual exam or the prelim, it sometimes helps to actually just underline the questions there. Okay, so let's get my pen working here. There we go. So it says farm 2. Okay, so we're only interested in the numbers, 240 and 348, and it's increased in yield when using modified cotton. So if you want to think of it as an arrow going that way, 240 going up to 348. Now you might be able to do that in your head, and if so, well done. But if not, we can use our calculator to work out the change first of all, because the first thing it says here, do a subtraction to work out the change. So I've got my calculator app here. So I just remind myself of the numbers. So it's going to do the bigger number minus the smaller number. So it's 348 minus 240. So 108 is my change. So I'm going to make a note of that in my wee space for calculation here. Okay, so there we go. 108 is my change. I'm going to divide that by the start, by the initial. Now it's gone from 240 to 308. So the start in this case is the smaller of the two numbers. You can see the 240. The other way to work out if it's an increase, it must increase from the smallest one to the largest one. So that's why it's the start. So it's 108 divided by 240 times by 100. And that's one that I definitely couldn't do in my head. So I'm going to go back to my calculator. Got my 108 in the screen. So I'm just going to divide that by 240. And I'm going to go equals. And then we're going to times by 100. And that'll give me the answer, 45. Okay. And you put your answer in. Usually in biology we're given the units, so you don't need to write the units in. The next question says, identify the variable altered in this. What did they change? What was different about the altered variable that they changed in this experiment? So as you can see, for each farm there was two different types, and so type's important, and there's cotton up above. So it actually says in the wee introduction, 3 farms had 2 fields of cotton, 1 of each type, and they were comparing original and modified. So we would say for that then our altered variable, in science we often call this the independent variable, but it doesn't mention that in this particular question. So the variable altered, the one that they've deliberately changed in other words, is type of cotton, and you must write that full. Please excuse my writing. Okay. In our skills booklet, the information to get about variables is in the first part here. Okay. So we see the independent variable, that's one they've just asked about. It's the variable that the experimenter deliberately changes. Its values are known before the experiment, and it tells you where it's found in the table or graph, and it tells you in the aim of the experiment where that will be found. For dependent variable, this is the variable that's measured. Its values are therefore only known at the end of the experiment. So this is our results, if you like. And it's usually found on the right-hand side of the table, or the y-axis of graphs. And in the aim, it comes afterwards the effect of something on something. Okay, so if it's the effect of temperature on enzyme activity, for example, temperature would be the independent variable, and enzyme activity would be the dependent variable. Okay. We also mention here what we call constant variables. To make an experiment fair, we don't use the word fair in biology, we use the word valid. It means the same thing. We keep our variables the same, we keep variables constant. And what we've listed here is just some examples of variables that you can choose. Okay. These don't always work. So for example, the study that we're just looking at is outdoors, it's out in a field. And our next question actually says, state a variable or suggest a variable that would have to be controlled to ensure validity. Okay, so in other words, to make it fair. And for this one, we could choose from our list, but obviously we couldn't choose temperature. You need to think about the specific experiment. This is out in the field. We can't possibly control what the weather does. So things like temperature, light intensity, CO2 concentration, which in other experiments might be reasonable suggestions, here won't work because in reality we could not keep those the same. Okay. So what sort of things do you think might work for this? What sort of things could be acceptable for variables? Well, we could have, think about the plants themselves first of all. We're obviously changing the type of plant. Okay, so think about the field. The farmer would have to sow the seeds in the field, so we could think about the mass of seeds that the farmer sows. We could think about the number of seeds that were present. Okay, that would be another one that we'd have to keep the same. It wouldn't be fair if we grew more seeds in one field than another. So we'd have to keep number of seeds or mass of seeds, perhaps more realistically the same. From our list, we could talk about pH. Now it wouldn't be pH of solution. In this example, it would be pH of soil, because that's what we grow the plants in the fields. We could also look at the one there that says time. Again, it would be time for reaction or time for process. It would be time for plants to grow. We need to give them time to grow so we can see how the mass has changed, how the yield has changed. Okay, so there's lots of good examples. You could also say things like area of field, size of field is also acceptable. We talked about number of plants or number of seeds, the spacing between the seeds. Okay, you want to have them at equal distances that we're growing, because otherwise competition would be a factor. So any of those would be acceptable answers. Okay, I'm going to choose, because it was on my list, so my pen keeps cutting out. So I'm having to use my hand now, so my writing's going to get worse. pH of soil. Okay. And if you're going to choose something like pH or temperature, if it was a different experiment, don't just say them on their own. pH of what, temperature of what is always a better way to do it. So the next question talks about controls. Okay, and this one is asking us, the plant with the original type of cotton, we regard that as a control field. And controls in science are used, again, they relate to validity, they make an experiment more valid. And again, in your skills booklet, you find a section on controls. So if I go on to the very next page, on the very next slide, slide 9, a control is something that's used to prove the variable we're investigating, so in this case the GM crops, is causing the changes, in this case the changes in growth, and not some other factor. If you're asked to describe what a control for an experiment should be, you would say carry out the experiment in the same way with the same apparatus, but replace whatever your factor is with the same mass or volume of number of whatever. Okay, so it's a bit different this one because it's an outdoor experiment, but and we're asked about why is the field with the original plants and not the GM plants regarded as a control. So for this question, how do we explain that? Well, it's used as a control. Well, think about what we're comparing here. We're comparing the mass. We have to have something to compare our modified plant. We're really seeing to try if that modification has made it grow larger or has increased the yield of the crop. So we could say to compare the mass or to compare the yields, gives us a comparison. It's to show that the difference in the mass or yields is due to the type of cotton. Okay, so you could come at it from that angle to talk about how the reason that the modified crop basically is larger, or to prove that the modified crop goes larger, we have to have something to compare it to. Excuse me, to show that difference. Or perhaps the one that makes most sense to me is to say to show that the modification is causing the changes, increases the mass, and that's the one that's most related to what our skills guide shows us. Okay, so it says the control is used to prove the variable we're investigating is causing the changes and not some other factor. So in this case, to show that it's the modification or the genetic modification that's causing the change in mass. Okay. Okay, so for that one I'll just put a wee text box answering. Okay, So to show that the modification is increasing the yield. That's cause and effects. The last one for question, for question 6 says describe how the reliability of these results could be improved. Now reliability again is a section in your booklet. I talked about this earlier on. Validity, accuracy and reliability in biology mean 3 different things. To achieve validity, we use a control, or we keep our other variables constant, keep them the same. To improve reliability, it involves repeating our experiments. Okay. However, what the SQE won't accept for an answer is just simply repeat the experiment. Okay. So the best way to do this is to try and relate it to the actual experiment. Okay. And if you think about what they have done here, they've set up just one field for the original type and one field for the modified type. Okay, and that makes it slightly less reliable. They've used lots and lots of plants, obviously, to get such a big yield, so that's a good thing. But in terms of the number of fields, they've just used one field for each type, the original and the modified. Okay, so the best way to describe your answer for this one would be, the best answer, sorry, would be to increase the number of fields, or to use more fields to grow the plants, and take an average of that, of the yields from each of the different fields. So for farms 1, 2 and 3, so for each farm and for each type, use more than one field to measure our results from. Okay, and this is quite important in science because it gets rid of any anomalies. What if one of these fields, for example, this very low one here, what if that 240 in the middle there had been due to, for example, something that had polluted the field, okay, or a lack of nutrients? So just to use one field for each type is less reliable. Okay. So again, I'll just type an answer in here. Okay, so I've not just said repeat the experiment. Okay, for this one you probably get away with just saying increase the number of fields used, or increase the number of fields planted with each type of cotton or something like that, but I've just given you a very kind of full answer there. Okay, so use more fields in each farm for each treatment, and then take an average is the best way to improve reliability. Okay, we're going to move on to question 8 now, which is the other question in this 2023 paper, which has got quite a lot of skills questions in it. But what it does, and we've seen the last question pretty much was all skills, sometimes they mix in some knowledge from your course. Okay, so for example, in Unit 2, Key Area 1, we learned about stem cells, and we found out how stem cells could be used to treat certain conditions, and can be used in research and things. So this relates to that Unit 2, Key Area 1. Okay, and this question is asking, describe the feature of stem cells that allows them to be used to be transplanted to replace blood cells. Okay, so we can use tissue stem cells, which can be transplanted to replace red blood cells, and describe what's the feature of stem cells that allows them to be used in the way. Okay. So a couple of possible ways you can tackle this. You might remember, firstly, that stem cells are unspecialised. Okay, so that's a good way to mention it. Okay, you can say they are unspecialised. You can say they can become specialised, so they've got the ability to specialise. Okay, remember our definition of cells. These are cells which are unspecialised, which can divide to self-renew and can become specialised. Okay, so it's the specialised bit we're interested in here, not the self-renew bit. You could also just simply say they have the potential to become different types of blood cells. Okay, so that's a nice, easy way to put it. So that's our knowledge question, but we really want to focus on the skills. So again, this one here. So this is one of these skills questions which really the guide cannot help you with. Okay, so in this particular question here, this is one that hopefully on the day you have a think about it and you try and work out the solution. It is kind of a pair question, and there's a section on pair questions which you could look over. We've got, sorry, my pen has decided not to work again, which is a bit annoying. There we go. Just undo that. So if you look here, that's an important word, okay? Those are our important numbers. And when I remember when I was doing my exams, this is quite something that I did sometimes with tricky questions. I pick out the key bits of information. What's the important numbers? But that per kilogram, as it says in your skills booklet, means for 1 kilogram, okay? So it says that to successfully treat a blood disorder, between 2 million and 4 million stem cells per kilogram, so remember that means for 1 kilogram, of the patient's body mass are required. Of course, our patient doesn't weigh 1 kilogram. Our patient weighs 78.5 kilograms, okay? So your sum here, how do we work that out? Well, it's the minimum number, okay? So we're not really interested in the 4 million. We could do it with 4 million, but the least we could use is 2 million, okay? So our sum is really a simple sum. It's 2 million. Okay, I'll write that out in numerical form. Okay, and we're going to times that, excuse me. It's not working again, I'm sorry, this. I will undo that, I'll type it. So it's going to be 2 million times 78.5. Now look in your question, okay? It doesn't give the units here, but it puts the word millions there, okay, on the right hand side. So because it's got the millions already there, we don't need to worry about the millions in terms of right now. We can just do 2. So our sum is a bit simpler. So it's just simply 2 times 78.5, okay? So again, we're going to put that into our calculator. You might be able to do that in your head. But you could always check it with your calculator even if you're confident. So 2 times 78.5 is our sum. And we get 157. Okay, so 157. The million is written for us, so we don't need to put all those zeros after it. So it's 157 in the answer slot there. And again, apologies. Can't seem to write that, so I will type in there. Okay. Okay, another skill we need to have in biology, and you've done a lot of that in class already, hopefully, is to interpret graphs. So this graph shows the survival rates of patients with a blood disorder who have received this stem cell transplant that was mentioned up above. It shows you the survival rate of individuals who've had this months after receiving the stem cell transplant. So it drops from, as you can see, 100 to just over 50%. Identify the survival rate of patients, it says, 24 months after receiving a stem cell transplant. Okay, so 24 months after receiving a stem cell transplant. So I go to 24 and I go up and I read it across and I get this point here on the graph. Okay, zoom in a little bit so that's clearer. And in this graph, hopefully it's obvious, there are 5 little blocks between 60 and 70 there. Okay, so there's 5 little blocks between that. So if it's 5 blocks between 10, one block, one little small square there on the graph paper must equal 2. So that goes 62, 64, 66, 68, 70, all the way up there. Okay. So the point that we've got on our graph is 64 there. Okay, so that's actually a nice straightforward graph question there. Okay. So 64, just reading straight off the graph. Describe the relationship, it says, between the number of months after receiving a stem cell transplant and the survival rate. So describe the relationship questions just tell us about the pattern. So this is not us explaining the graph, explaining what it shows, it's just telling us what we see, what the pattern shows. So for this kind of question, you really have to mention what's happening down both axes of your graph there, okay? And as you can see, as the number of months after receiving a stem cell transplant increase, the survival rate decreases. Okay. So how do we put that into text? Okay. Well, if you go to your skills booklet again, and we go to the section on describing the graph, You'll be asked to use values from the graph. You must use data points from both axes at each change in the relationship, so any increases, decreases, or levelling off. And remember to name the values and units from both axes as part of your answer. So the only unit we've got is on our y-axis, and that's percentage, so I must remember to put percentage in my answer. So we're going to say, as we can see over the course of this study, there's only one change, only decreases. And that's why this is a 1 mark question. Sometimes if it decreases then levels off, or increases then decreases, it's for 2 marks. But for this one, it's very simple. We're going to say for this one, as the number of months after receiving a stem cell transplant increases from 0 to 60. Okay, so that's going from left to right at the bottom. The survival rate decreases from 100. And we can see this number here. It's halfway between 50 and 52, so that'll be to 51%. Okay, so your model answer would look like this. So I'll smooth that down so you can see it. Okay, so as the number of months after a stem cell transplant increases from 0 to 60, and notice there's no units for that, just 0 to 60 months obviously, the survival rate decreases from 100 to 51%. Okay, and the last one again, they're mixing up a bit here. They've got some skills questions and some knowledge questions. So question D there is knowledge from your course. Other than tissue stem cells, name another type of stem cells, and you should know that there are tissue stem cells and there are also embryo or embryonic stem cells, as you might have learned them as. Okay, so we'll put a wee final answer in there. My pen's back working again, so I'll try my... Excellent, okay, so embryonic stem cells. Okay, so hope that has helped. I would encourage you very much to go to, certainly before your prelim, to not forget about the skills. Okay, it's very important that you're using the processes we talked about earlier to learn the knowledge, to check your knowledge and to test your knowledge at exam level, but it's as important to have a go at lots and lots of skills questions and practice them, and please use the skills advice booklet to help you with that. Okay, we'll leave it there. Many thanks.